In this lesson, we are going to have a short tutorial on amino acid structure and how they are involved in protein synthesis. We also look at the various classes of amino acids, which is based on their chemical properties and also how they behave in a solution of different pH, that is the isoelectric point of amino acids. Don't forget to like and subscribe to the channel after watching so they can receive every update or post on this channel. So amino acids are basically the building blocks of protein, and they are merged millions to form different proteins. Two or more amino acids links together to form a peptide, and the peptide also interlink to form protein structure. Structurally, amino acids have two functional groups. They have an amine group and also a carboxylic acid group. Amino acids have at least 4 different atoms and a maximum of 5 different atoms in a structure. Every amino acid has a nitrogen atom, a hydrogen atom, a carbon atom, and an oxygen atom. Other amino acids have sulfur atom present in them. The amino group or the amine group and the carboxylic acid group of amino acids are bonded to a central carbon we call an alpha carbon, which is also bonded to a hydrogen atom and an R group. The alpha carbon has 4 different groups bonded to it, and that defines the chiral nature of amino acid. Except for glycine, all amino acids are chiral because they have a central carbon, which is the alpha carbon, this carbon, bonded to 4 different groups. Amino acids condenses or engages in the condensation reaction to form peptides through a peptide bond formation. Peptides intercondenses to form protein. So you have two amino acids, let's say an amino acid 1 and an amino acid group 2. These two amino acids undergo a condensation reaction with the loss of water to form a dipeptide through a bond we call a peptide bond or an imide bond. In peptide bond formation, the oxygen from the carboxylic acid of one amino acid and 2 hydrogens from the amino group of and the amino acids leaves to form water, whereas the nitrogen and the carbon or the carbonyl group forms a bond we call a peptide bond and or an imide bond. So in every amino acid or peptide, the number of peptide bonds or an imide bond present in there is equivalent to the number of amino acids decreased by 1. So we have peptide with 2 amino acids with 1 peptide bond. So in peptide bond formation, when the carbonyl carbon atom loses an oxygen atom and the second amino acid contributes 2 hydrogen atoms, a dipeptide bond forms joining the 2 amino acids. So let's say, for example, if you have these amino acids, the oxygen from the carboxylic acid group of the first amino acid and 2 hydrogens from the amine group of the second amino acids interjoin or condenses to form water, whereas the 2 atoms join together to form an imide bond. So for example, if you have, let's say, a polypeptide like this, the number of amino acids present in there can be found by counting the number of the side chains. So we have side chains 1, side chain 2, 3, 4, and 5. So since we have 5 different side chains in this polypeptide, it is clear that this polypeptide has 5 different amino acids. It can also be found by counting the number of nitrogen atoms present in there. So we have first nitrogen, second nitrogen, third nitrogen, fourth nitrogen, and the fifth nitrogen. So we have 5 different nitrogens present in this polypeptide. This means that this amino acid or this polypeptide has 5 different amino acids. To determine the number of peptide bonds present in there, between the first amino acid and the second amino acid, there exists a peptide bond. Between the second amino acid and the third one, there's a peptide bond in there. The third and the fourth, there's a peptide bond in there. And the fourth and the fifth, there's a peptide bond in there. So between serine and leucine, there is a peptide bond in there, or an imide bond. And between leucine and threonine, there is a peptide bond and imide bond in there. Between serine and threonine, there's a peptide bond in there. and between the serine and valine there's a peptide bond in there. So for every polypeptide bond, the number of peptide bonds or the amide bond is just the number of amino acids decreased by 1. So let's see what accounts for the difference or the variations in amino acids. So every amino, as we said earlier, has different R group, and they have constant amino group, carboxylic acid group, a hydrogen atom which is bonded to the central carbon, and the differences in the R groups defines the different amino acids or the type of amino acids or cause the variation in amino acids. Let's say if you have an amino acid with the R group which is H, this amino acid is glycine. This H differs in valine and it gets replaced by an isopropyl group. So here is the central carbon, which is the alpha carbon with the amino group, the carboxylic acid group, and the hydrogen. And in valine, there is an isopropyl group, which makes it different from this. In the same way, if you take amino acid cysteine has the R group, which is a sulfhydryl group bonded to a methyl group. So this one is different from this. So the differences in the R groups causes variations in the amino acid. We take every central amino acid with an alpha carbon in there. It has an amino group, a carboxylic acid group, a hydrogen atom, and an R group, which define the properties and causes variation in different amino acids. So let's see how we can class amino acids. So amino acids are classed based on different structures of the R groups. So based on the R group, we have 4 different classes of amino acids. Other books say 4 and subdivide, or 3 and subdivide the 4 and 3 into 1. So based on the R groups of amino acids, you can have a non-polar and neutral amino acids. You can have polar and neutral amino acids. You can have an acidic and polar and also basic and polar. So the acidic and polar amino acids are the charged amino acids. So let's have an example: non-polar amino acids with aromatic ring. So these are the 3 main amino acids with an aromatic ring. We have a phenylalanine with a benzene ring, which is neutral. We have a tyrosine with a benzene ring, which is quite non-polar, and also a tryptophan with a benzene ring and a quite non-polar. The tyrosine and tryptophan has oxygen atom and nitrogen, which confers some kind of polarity in there, but it's quite negligible when it comes to this consideration. So other polar amino acids with polar functional groups means that they have an electronegative atom present in the side chains. So they may have an oxygen atom, let's say nitrogen atom, or even a sulfur atom in there. So in the side chain of some amino acids, you can have an amide group present in there. You can have a hydroxy group present in there, or sulfur with a hydrogen present in there. So these groups present in side chains makes up some amino acids polar. So for example, serine has a hydroxy group as part of the side chain. 309 has hydroxy as part of the side chain. Cysteine has a sulfhydryl group as part of the side chain. A tyrosine has a hydroxy as part of the side chain, and also asparagine has an amide group as part of the side chain, and also the glutamine. So these 3 groups present in side chains of amino acids makes them polar. Let's see amino acids with charged side chains. So charge means that they can either be minus or plus charge. So amino acids with plus charges are mostly the ones with an amine group as part of the side chain. Since amine, let's say from ammonia is a base. If you have an amine present in a side chains, it makes the amino acid a basic amino acid. If you have carboxylic acid group part of the side chain, it makes the amino acid an acidic amino acid. For example, we have this one, leucine. Leucine has one amine group present in the side chain. This makes leucine a basic amino acid because ammonia as a functional group is a base. Arginine has about 3 different amine groups present in the side chain that makes it some kind of basic amino acids. Histidine has an ammonia or an amine group what is charged that makes some kind of basic amino acid. Acidic amino acids are the ones with carboxylic acid groups as part of the side chain. So aspartic acid and glutamic acid are the non-acidic amino acids which are polar. So let's have this one, the acid-base properties of amino acids. So amino acids have acidic functional group and also basic functional group. The amino group of amino acids from the word ammonia acts as a base in solution, and also the carboxylic acid group of amino acids also behave as an acid in a solution. So because of these two functional group, amino acids being both acidic and basic, we describe them as amphoteric compounds because they have both acidic and basic properties. Let's say if you have a solution of a higher pH as a basic solution, the carboxylic acid group of amino acid will lose a hydrogen and form a carboxylic ion and assume a negatively charged net compound. In other words, if you have a solution of more acidity, the amino group of the amino acid will accept one proton from the acidic solution and assume a plus charge. So they can behave as both acid and base in solution. So let's say you have a neutral pH, neutral pH of 7, where we must have water can act as both acid and the base. Here, in the neutral solution, the amino acid will act as both acid and base, and it does so by abstraction of the proton from the carboxylic acid group to the amide group of the same amino acid to exist as dipolar ion, which is of negative ion. We have amino acid with two different ions, a plus and a minus charge with a net charge of 0. This ion is what we call a zeta ion. So amino acid shares some basic properties with salt. For example, some amino acids can form crystals. They have higher melting points. They are soluble in water, of course, but are not soluble in a hydrocarbon solvent, because these solvents are mostly non-polar solvents, but amino acids are polar. So let's see how amino acids behave in solutions of different pH. Neglecting the structures present in the side chain, amino acid can behave as both acid and base in solutions based on the properties of the pH of the solution. Let's have a solution of high acidity, that is a lower pH. In a lower pH solution, the amino group of the amino acid will accept a proton and exist as a cation, because the amino group is more like a basic functional group. In a neutral solution, that is about 7, the amino acid can both accept a hydrogen from the neutral solution, also donate a hydrogen in the neutral solution, because in a neutral solution there is some kind of amphoterism. It has both acidic and basic property like water. Water in this solution can both donate a hydrogen to the amino group and also take a hydrogen from the carboxylic acid group, making it exist as a dipolar ion with a net charge of 0, neglecting the charges on the R group. In a high pH of an alkaline solution, the solution will accept proton from the amino group and make it neutral, and also accept proton from the carboxylic acid group and make it ion. This means that for every amino acid, it can exist in at least 2 different ions. So this one, if you have this in a lower pH, we have a plus charge state. In a neutral pH, we have a 0 charge state. But in an alkaline pH, we have a negatively charged state. That's the various state of amino acid in solutions of different pH, neglecting the charges on the R group. Now, if we have this, let's say you have an acidic solution, as we said earlier, the amino acid will accept proton and exist as a cation. In a basic solution, it will give out a hydrogen to hydroxy group in the solution, that is in a basic alkaline solution, and exist as an ion. But in a neutral solution, it will exist as dipolar ion. Dipolar ion means that it has both negative and positive charge. Now, the pH at which the dipolar ion of different amino acid forms are different. So the pH at which the net charge of the amino acid is formed or is 0, or the pH at which the dipolar ion is formed or the pH at which the zeta ion is formed. That pH is what we call isoelectric point. So the isoelectric point of every amino acid is the pH at which the charge on the amino acid is zero, or the different charges, the negative and positive charges, are evenly balanced. So let's see